we've reached the final talk of the summer school. I hope everyone's had a good time. And for the final talk, I'm pleased to welcome Emma Robinson, who's head of biomedical computing in King's College London, and is going to talk about precision neuroimaging by Explainable AI. So over to you.
All right. Hello, everyone. Thank you for inviting me. So today I'm going to talk about explainable AI, but from a very different angle, focused on applications in neuroimaging. And in order to motivate that, I need to start by giving some background as to why clinical diagnosis of neuropsychiatric disorders is such a hard problem. To give a definition, what I think we're really all aiming for in precision neuroimaging is what we'd ideally like to be able to do is to take some individual subject, where this is actually my son Ash, volunteering for the Developing Human Connectome project as a baby. And from these healthy babies, we acquired multiple different MRI scans, so we can visualize their brain shape, we can visualize their microstructure, so how, indirectly infer how the neuronal cells are connected and how they communicate with each other to confer brain function and encode different aspects of cognition. And what we'd like to be able to do is to combine this imaging data with demographic information, clinical records, perhaps genetics, and push these through learning models and simulations in order to get out tailored predictions and prognosis, for example, what their risk of autism might be. Similarly, at the other end of the spectrum, what you see during healthy aging is that these brain tissues start to atrophy, which is most clearly visualized as the most salient feature of this is that the fluid-filled spaces in the brain, so the ventricles and the outside CSF, are getting bigger. And what you want to know is that for any given individual, whether their brain atrophy is following the trajectory of the normative population, or whether it shows signs of accelerated aging, and that would give you some indication of whether they might have early signs of dementia. And the reason why this is hard, well, there's a hierarchy of reasons. The first one is that we need to be transparent that neuroimaging data is very low resolution relative to the cellular processes that are driving neurodevelopment, cognition in disease. So while it gives us really valuable information because we can look at what's going on in vivo, in a live human, at a scale where we can observe what's going on across the whole brain, which just wouldn't be possible. We can't model whole brains at the cellular level, the amount of neuronal cells, there's billions of them. But it's not telling us what's going on at the molecular or cellular level, which is where these diseases are starting. So we're already going to be picking things up at a later stage. It's also really expensive, so we can't densely sample within each individual, unless you happen to be Ash, who is probably the most densely sampled child in London. So he keeps being volunteered. He keeps volunteering himself for MRI scans. So we can look at their individual brain. We can look at his individual brain growth trajectory, but in general, we can't do this for every single baby. So we need to infer these patterns cross-sectionally, but then we have a real big problem because every single person's brain is very different, which perhaps might not seem all that surprising because we all have very different personalities and we think very differently, but we need some way of being able to compare across individuals in order to define what's normal. And this is true both in terms of the brain shape, but also in terms of their functional organization. The biggest source of variability is the cortex. So your cortex is the layers of grey matter tissue at the outer surface of the brain. Your cortex is one of the areas of the brain that's most evolved relative to non-human primates, which implicates it in most higher order cognition. And the grey matter tissue is where you find the cell bodies, so the thinking parts of cells, whereas the white matter is the connections between these grey matter regions. And what happens during neurodevelopment is these cells, the neuronal cells, they start in the center of the brain around the ventricles, and they migrate out radially to form the cortex, the 6 layers of the cortex in an inside-out fashion. And when they get to the cortex, they form hands. So you form these intercortical connections, which cause the cortex to grow at a much faster rate than the tissues below. And this causes biomechanical tensions, which cause the cortex to fold. And this doesn't happen uniformly across the cortex, but happens in a well-characterized schedule, starting with the central sulcus and the sylvian fissure, so primary folds, and then progressing on to secondary and tertiary folding around 31 weeks gestation and then just before birth. These primary folds are thought to be mostly controlled through gene expression. And then this way, they're largely conserved across species and across humans. But as you progress through development, this process becomes more and more unstable and potentially susceptible to random differences in the rates of cellular proliferation, for example. And what you find then is that the later areas of the brain to fold show much more variable patterns of folding across individuals, even genetically identical individuals. So this can be seen for these 3 healthy adults here. So they're perfectly healthy. These blue and yellow maps represent what happens if you inflate this cortical surface to a smooth convex hull. So the yellow is the tops of the folds of the gyri. The blue is the depths of the folds of the sulci. And in the frontal lobe, which is one of the late developing areas and the area of the brain that's responsible for much of your complex cognition, so your working memory, your reasoning, these areas look and fold completely differently in one individual to another. And then to make it even more complicated, the cellular organization of the brain, so the tendency of your neuronal cell bundles to cluster into areas which have specialized functions, this is dissociated from shape. So you can't locate these areas from your cortical folds alone. And these show their own varying patterns of cortical organization. And then your disease-specific variation or your phenotypically specific variability is much more subtle, and it's occurring on top of all of those sources of variation, which means that it's really very difficult to tell from neuroimaging data alone whether an individual has any form of neuropsychiatric disease. And so this creates a lot of challenges because you need to be able, so in order to be able to diagnose whether someone has some form of neurological disorder or neuropsychiatric disorder, you need to be able to compare, you need to define what is normal for them. So you need some way of comparing against populations. And that means you need some way of proposing some simplified model or some way of mapping from the space of one person's brain to another. So classically, how this was done was that, before the days of AI, we had to use classic image processing techniques. And so we would take the shape of an individual person's brain and deform that shape in a smooth and continuous way until it more closely resembled that of some population average shape. And we would do that for all of the brains. And then you would assume in this space that those brains completely overlap, so that you can compare common cortical areas across populations in order to statistically compare between groups. So this has taught us a lot about the general population trends of different neurological disorders. So we know that Alzheimer's, for example, starts in the hippocampus and then progresses through the temporal lobe. We know that this is a recent study by my student Mariana. You know that if you compare the cortical growth patterns of fetuses to preterm babies, those areas of the brain that are growing latest during gestation are less well developed or are growing less well than if they were developing healthily in utero. But it completely ignores the scale of the variability that we know exists across humans, not to mention the fact that if you're performing all your alignment based on shape, and you know that the cortical areas don't line up with the shape, then you're not doing a good job of comparing the cellular or the underlying cytoarchitecture across individuals. And so what happens then is that if you're trying to build some normative model of how you expect the population to develop either during neurodevelopment or healthy aging, and your objective is to say that outliers from that model represent signs of pathology, then you are unable to detect these subtle signs of disease because they're hidden amongst all of this image measurement or experimental noise. So actually, when you build these normative models, so this is again these circle depth maps, and on the x-axis you've got weeks of gestation, and on the y-axis you've got the circle depth, so the depth of the folds. And these outliers just happen to indicate a small subset of individuals who happen to have an atypical pattern of cortical folding at this location. So they have a rare gyrus where most individuals have a sulcus. So classical image processing doesn't work. What we need to be able to do is build models that tell us what the normative development should be relative to the anatomy of that individual. So does AI offer a potential solution? And the short answer to that is obviously yes, otherwise I wouldn't be here. But only, and I think this is particularly important for brain, but I would say this is uniformly true for biomedical AI, only if you embed a deep understanding of the underlying sources of variability into the design of your model, which is why I'm giving you all this background. So you can't just, you never have enough, or you'll rarely have enough. I think in the case of the brain, you'll never have enough imaging data to model all of the possible sources of variability. And therefore, if you build simple models that try and encode this in a completely unconstrained way, then you're never going to be able to generalize or, derive biologically plausible or human interpretable explanations that will make them useful for clinical use, in my opinion. So why would you want to use deep learning? And that's because modern deep learning networks can be trained to be transformation invariant, which means that they can learn to model those residual sources of variability that you just can't account for through smooth diffeomorphic image registration or any other pre-processing technique. And so, for example, the reason why this is true for convolutional neural networks is that they're not comparing images, assuming they're perfectly overlapping. Instead, they're learning these filter kernel representations, where these filters are learning to be sensitive to different types of texture or object in the image. So as you all probably know, convolutional filters they're usually about 3 by 3 or very small filters that get translated across the surface of an image. And the parameters of those filters are being optimized during backpropagation through your network, such that over many layers with downsampling as you go through, you're learning a hierarchy of filters that learn to recognize more and more complex textures in the image. For example, your early layers are learning edge detectors. You then start to learn more complex textures. And then once you get to the end of the network with all of the downsampling, you start to be able to recognize individual objects. And so you're comparing images in terms of their relative distribution of these textures or objects in the image. The problem is that in their simplest form, deep networks are black boxes. So in learning these filters, these networks are training millions of filter weight parameters through complex nonlinear interactions that aren't really human interpretable. And so even if you are to look at what the individual filters look like or what objects are activating them, it's not really giving you a clear explanation of exactly how they're combined for the network to make any decision. And this is a real problem for medical AI, because what can happen is the networks just learn to cheat or shortcut. So there are many examples where you can see that the networks aren't learning to model the biomedical problem. They're actually fixating on some annotations that some clinicians have put on their scans. or they're cheating because the data set is not stratified. So predominantly, most of your samples come from healthy individuals, and there's just a small subset represented from the disease class. And then if it just classifies all of them as healthy, then, numerically, it's doing a really good job. The other problem is bias. So models, the clinical care is already biased because clinicians themselves will probably see a far higher proportion of people from representative of their own community. So in this country, we'll be mostly seeing examples from whites, Caucasians. So in many cases, your label data is biased because it's coming from trained clinicians where it's biased, but then the networks will encode that bias and perform preferentially, or perform less well on protected groups, and that then becomes a major problem. So AI algorithms that are rightfully held to a higher standard in terms of their expectations of how they perform fairly across populations. And in order to be able to work out whether it's doing what you want it to do, you need some sort of human interpretable, ideally visual explanation. So historically, what people did initially was they would train discriminative networks from classification or regression, and these would give you a prediction which you could then push back through the network, and you can track back through the network trying to look at which features in the image were most important for driving that decision, and then you can get some form of heat map that will give you, or saliency mass, which will tell you which features are most important. This was a good start, but it's got lots of problems. First of all, these are inherently low resolution because of all the downsampling that happens through for a convolutional neural network. The resolution at the output is very low. I think, but more importantly, if you're just training networks to perform classification or regression, then they only have to learn the most salient features in order to perform that task. So if we go back to this example that I was giving at the beginning about Alzheimer's, we know that the most salient feature is the increase in the fluid filled spaces of the ventricles at the center. So a classifier network is just going to focus on the ventricles, and not even all of the ventricles, just the top left corner here, and it's going to ignore all of this potentially clinically important pattern of cortical atrophy around the outside because the cortex is too variable across individuals and it's too hard to model it, so it just ignores it. So a better approach, one that people are increasingly using, is to train generative models that can be conditioned to give you a visual explanation for why an image has a particular class. So one of the first papers that I remember doing this particularly well was done by Christian Baumgartner in 2018. So he trained a generative adversarial network to generate a difference map which will transform an image of class 1, for example, diseased, back to a healthy state. And the reason why this works is best seen through the simulation of this paper. So what he was hypothesizing is perhaps you could imagine a case where you might have healthy and diseased individuals, but within the disease class, there are many different subtypes of disease, and perhaps some of them share some common features, but there's always some patterns of disease that are slightly different between the two. If you train a classifier network to detect disease versus healthy, it just needs to focus on what's consistent between the two populations and not what's different. But if you train a generative network, which has to learn how to make these images healthy, it has to remove all signs of disease. Otherwise, it'll still be able to tell the difference. So in that case, you get a comprehensive explanation for everything that's a comprehensive feature map that's going to pick up all signs of disease in that image. The problem is that on its own, as I said, we don't have enough imaging data for it to be able to model that entire space of variability for the human brain. And so after that, my postdoc, Cher Bass, took this idea and expanded on it by incorporating this understanding that we know that most of the variability in these images is actually natural variability that we don't care about. and trained a generative network to learn two latent spaces. One larger content space, which is learning to model the space of natural brain variability, and another smaller attribute latent space, which is a VAE, which is learning to model the disease-specific or the phenotypic variation. And you've got regression or classifier modules on each of these networks that's pushing out the disease content out the disease information out of the content space and into the attribute space. And then when you train, you can learn to change the class of the image by retaining its content latent vector, but swapping out its attribute latent vector for one of an opposing class. And then you get essentially a better explanation or a more comprehensive explanation that's picking up on all signs of disease. So here, again, we're looking at Alzheimer's. This is the ground truth. So you've got some individual who was scanned twice longitudinally, during which time they progressed from a state of mild cognitive impairment to Alzheimer's. So this disease map is basically summarizing what it was that clinically led to a decision that they had converted. And ICAM picks up on all signs of disease, not just the ventricular atrophy, but expansion, but also the cortical atrophy. Whereas VA-GAN, which is Christian Baumgartner's version, struggles a lot more with the amount of data it has available. And these post hoc explanations are incredibly coarse and non-specific and not that human interpretable. And you can take that further from classification to regression. And because you've got a variational autoencoder, you can interpolate through the latent space to learn continuous explanation for what would happen if you gradually change from one class to another. So this is healthy aging. So this was really positive, but it's not taking into account everything we know about cortical modeling. So it's ignoring a crucial property, which is that the cortex actually is not a three-dimensional object. It's a two-dimensional manifold embedded in a three-dimensional space. And we know that surface modeling of the cortex is extremely valuable because there's been a large body of classical image processing-based analyses with with big impact results, some of which I've shown in these previous slides, that show that analyzing cortical imaging properties on the surface significantly improves the precision with which you can compare across individuals. So we know it makes sense to model the cortex as a surface. Maybe this is a good visual explanation of why. So these are the brain functional activations, and I've said that the cortex is made up of cortical areas. So you can see that the yellow, when Chen was showing these maps of time series, I would argue very strongly, and some of the results that I'll show in a bit later might evidence this, that it makes much more sense, rather than to parcellate the brain with all the difficulties that there is in commonly identifying common areas across individuals. Instead, just treat your brain functional activations as a moving image across the surface. And then you can capture the spatial autocorrelation or the topography of the signal as it evolves through time. The problem with doing this is that generalizing deep learning methods to non-Euclidean domains is actually really challenging, and that's because they don't have any global coordinate system. So I don't want to bore with you too many details, but essentially the reason why CNNs are so powerful is to do with the translation equivariance property, which means that basically for Euclidean domains, it's very simple to define a regular Euclidean filter grid, which you can then translate across the face of an image, which will then generate an output which is also mapped to the convolutional, which is also mapped to a Euclidean domain. And that output is translation equivariant. So it doesn't matter whether you translate the image and then convolve it, or you convolve it and then translate it, you still get the same result. With surfaces, you have no global coordinate system. So your coordinate system is defined, you have a local coordinate system defined in a tangent space, which is, relative to your starting position. And there's no way of defining a regular meshing of the surface, which is equivariant to the rotational orientation of the image that you present. So the convolutional operation, if you rotate a filter over the surface, the orientation of the filter is path dependent. So if you started with a filter here and you translated it over the top of the sphere, it would flip upside down. And if you went around the equator, it would flip left to right. So the convolution operation itself is path dependent. And then that means that when you present an image on a sphere to a surface convolutional neural network, if it's not aligned, then it won't easily learn a transformation equivariant representation. It's just really hard to do this in a computationally efficient way. You can do it in the spectral domain. So the convolutional theorem says that the convolution of the Fourier transform of the convolution of a filter and an image is equal to the product of their Fourier transforms. And so you can generalize Fourier bases to any domain. So for a sphere, you have spherical harmonics. For a general graph, you have the Laplacian spectra. But this ends up being extremely computationally heavy. So what happens is you will have perhaps heard of graph convolutions. You might have heard of point nets. There's a whole bunch of different ways that people have tried to generalize convolutional operations to non-Euclidean domains. They're each specialized to the domain of interest, and each one of them tends to make some form of approximation. So you don't get all the properties that you would get from a Euclidean convolution. So to work out which one of these was most important for brain imaging data, we benchmarked these on the task of predicting how old a baby was when they were scanned from their imaging data, or segmenting their brain into cortical folding-based regions, and came to the conclusion that the data is sufficiently complicated and noisy that you don't want to take a hit on how the expressiveness of your filters, which meant that graph convolutional type approaches weren't ideal. But you want to maintain as much rotational equivariance as possible. Whatever you do, you're making a compromise. If you work in the spherical domain, it's so computationally heavy that you can't design complicated or intricate networks or do anything particularly sophisticated with them. Nevertheless, you can do some pretty powerful things. So some collaborators of mine at King's took graph convolutional neural networks and put them into a no new U-net architecture, and use them to segment focal cortical dysplasias. So cortical dysplasias are very, very subtle, tiny lesions in the cortex, which tend to be the site of drug-resistant childhood epilepsy. Very often, you cannot see them at all on structural MRI. Often, the clinical workup involves many different types of imaging data. They're really difficult to find. And while you can't find them, these children, their brains are getting more and more damaged through the seizures. So what they did was they trained a network to segment it. They found that it performed better than clinicians, and now they're trying to work this into clinical practice. So these work. And you can use this to get towards these deep normative models that I was saying that we needed to aim for. Hello?
I was just wondering how the children are identified, like to start scanning them.
These children, so they have a big project called MELT, and they've acquired scans that have been anonymized of children across the entire world. So they've just acquired data. So these children present with drug-resistant epilepsy. And then there's a path towards, most often the treatment will be surgical resection of that lesion.
Okay, so you need to find where the lesions are.
They're trying to find where the lesions are. And if you can, then quite often, if you remove them, the outcome is good.
Thank you.
So you were saying before that one of the disadvantages is the complexity, computational complexity for this very ones.
So it's STCNN. That one's a spectral. That one is a spectral approach. So you have to actually go. So you start with a spherical harmonics, and then you're in the space of 3D rotation. So you have things called the Wigner D bases. So it gets really, really computationally heavy. So you can train a network, but if you wanted to build a complicated generative network like the ones that I'm coming to, it just becomes really computationally heavy. So I don't know anyone who's using it, actually. So most people, the graph convolutions, they are the original graph convolutional neural networks. They use filters that look like bullseyes, like concentric. They can't learn complex textures. So you have other methods that will do spatial templating, and then they look exactly like a normal convolutional filter, except for they're hexagonal, and you have free learning of all the weights, but then they lose rotational equivariance completely. So everything is a trade-off.
But you train bunches, right? Bunches of the images, or
We train on whole images projected to spheres using these different things. So mostly we use something known as a MoNet feature, which was one of Bronstein's papers from like 2017, I think. And that uses a mixture of Gaussians filter, where the Gaussians are lined up with the polar coordinates of the sphere or the surface, local polar coordinates. But increasingly, we're moving to vision transformers, which is what I'll talk about in a minute because of all these trade-offs. But I mean, that being said, convolutional neural networks are much more parameter efficient. So for low data regimes, they are more they're better for generalization. Yes, so we ended up going with MoNet filters because they're reasonably expressive and you can train them to be rotationally equivariant. And we've done quite a lot of image processing based things with them. And these normative models. So these were MoNet convolutions. So here it's the same idea as ICAM. You learn a difference map, and that difference map will smoothly or continuously age a subject forward and backwards in time. We cheat slightly by training on a data set where we have preterm babies' first scans, and we kind of assume that they're pseudo-healthy, which is not accurate. But at this point, we didn't have the fetal data, so perhaps we'd need to retrain. But you're training these models to simulate healthy neurodevelopment. And in that space, then you're moving away from this because you have a personalized normative model that tells you what that individual's brain should look like if they were healthy. If you test independent regression network on predicting age from these simulated scans, it does really well. And if you compare the simulated healthy scans from the ground truth follow-up scans for preterms, then you do a better job of predicting their outcomes at 18 months. So this is really promising. But at the same time as this, knowing all the problems that we had with convolutional neural networks, Simon Dahon, who was an amazing PhD student who I had, who's just left and gone to work in brain-computer interface technologies, and he's helping paralyzed people walk. He did all this work on surface vision transformers. So the great thing about transformers is essentially they can be they're domain invariant. So as long as you can find some way of patching up or tokenizing your data, you can apply them to any domain. So the concept of vision transformers, the paper originally came out in 2020. Transformers traditionally were used for natural language processing. And the reason why they were so powerful is because they learn long-range associations between large bodies of text so that you can know that one word that comes many, many sentences after is referencing, or one phrase is referencing back to some sentences a long time ago. So it gives you long-range context. And Dosovitsky et al., what they realized was long-range context is actually really useful as well for scene understanding. So convolutions, they're learning these very, very localized filter representations, and that makes them super powerful and parameter efficient, which is really powerful for generalization, but they don't really have an understanding of the image per se. It's picked up from looking or trying to identify individual objects. So there's a good example from the capsule paper from Jeff Hinton that says, well, it will classify an image as a face, even if you've got the nose and the mouth and everything just in completely the wrong place. So it doesn't really have global contextual information. But if you train a vision transformer, then you can get long-range context. And you do this just by patching up your image. So you take your image, you split it up into regions, you flatten those regions, you linearly project them onto some token domain, and then you just push it through a standard transform, the same thing that you would use for language processing. And the reason why they're so powerful is they learn these self-attention representations. So they've got these huge weights matrices, which is why they're learning more parameters. And these weights parameters are learning this self-attention representation, which is telling you how important any given token is, how important all the other tokens are to some particular token, and that relationship is directed. So from language, I think it's easier to understand this in the concept of language. So with this sentence, Evan's dog Riley is so hyper she never stops moving, it's more important to know that the subject of the sentence is the dog than actually know what the name of the dog is. So the importance of Riley to dog is much greater than dog to Riley. But it's also important for modeling neuroimaging data, as I'll come back to in a few seconds' time. So this is learning the importance of these different image patches relative to each other in order to understand what the image is about. And what Simon realized was that basically, as long as you can find some way of patching up your domain, then anything can be pushed to a transformer. So he took the same idea, projecting the cortical imaging data to a sphere, patched it with the regular triangular mesh known as an icosahedron, did the same thing, pushed it through a transformer. The problem with transformers is that they're really data hungry because they learn way more parameters because they've dropped all these inductive biases that you have for a convolution. But you can, as Chen was saying, use these self-supervision practices. So this is a masked autoencoder that she also showed on her slides for imaging, to learn, to pre-train those self-attention mechanisms so that it really has a deep understanding of the underlying data. So in this case, you patch out, you remove huge amounts of the data, you push it through a thin but deep encoder, which has to learn really sophisticated self-attention mechanisms in order to then fill in all of these missing patches and reconstruct the image. And when you're doing this, I think it's most striking when you do it with fMRI data. So these are 3 different frames in time of fMRI data. And when you train the masked autoencoder, you remove 90% of the patches. So there's hardly anything left. So the only way that it can learn to reconstruct these is to have a really deep understanding of what the underlying spatial autocorrelation of the cortical aerial topography is. So it needs to know that if this area is firing up and this area is firing up, then this area must also be firing up because these 3 regions are connected and are involved in a specific functional network, which is encoding a specific stimuli that this individual is watching during their fMRI experiment. And this means that it has to have some real understanding, or it has to have some underlying model of the functional dynamics in order to be able to do this. And then through doing this, you then get AI networks that strongly outperform convolutional neural networks. So this was benchmarked on a regression task, so predicting how preterm a baby was when they were born from their term age scan. And it performs in a translation invariant way, so you don't have to align the data at all, and it'll still perform really well. But more importantly, transformers put out a human interpretable visual explanation for why they made their decision. So you can visualize the attention maps. So for a discriminative task, you always have a prediction head, a fully connected neural network at the end that makes your prediction. And into that goes your classification token. So you can look at the attention of all your image patches to that classification token, and it will tell you what it was looking at in order to make its decision. So here, and you get a tailored representation for each individual. So for these babies, what you know is over this late period of gestation, the folds are deepening, and this area of this is the motor sensory cortex is maturing. So the myelin is what coats the neuronal cells in order to improve their efficiency. So it's increasing a lot over this period of time. And the networks, although it's quite a coarse scale tokenization, the networks are focusing on these areas. So it's giving you an explanation of why the baby was given the age that they were. So this is moving us closer to a interpretable explanation that is parameter efficient in the sense that if you're modeling cortical, especially fMRI, if you're modeling functional dynamics on a surface, then you've got there's far few surface points than if we were modeling in a 4D volume. So you're right now starting to be able to do much more powerful things. But I would argue that we need more than just visual explanations. We need to use our imaging data to give us a mechanistic understanding of what it is that's driving driving the classification of some particular disease phenotype or something. So you can't just say, oh, well, I think this baby has autism. Why? What is it about the brain imaging data? What is it mechanistically that's going on that explains that disease? Because if we don't have some sort of explanation, then how could we ever develop any form of useful treatment? So this is the direction that we're moving in particularly. So the first example of this is still work building on this vision fMRI or surface masked autoencoder. So here, what you can do is you can start to model what a person is thinking. And the first step towards doing this is generalized decoding. So a decoding task with fMRI is you want to be able to predict what an individual is looking at or what stimuli they're receiving purely from their brain activity. So in these tasks, usually these people are experiencing some sort of naturalistic stimuli in the scanner. So in this case, it's a movie watching task. They're watching lots of relatively short parts of movies like Inception, and you're recording their brain activity during that. And what people classically do is they ignore the spatial autocorrelation or the structure of the signal, and they just try and treat each fMRI signal, the time series is independent. And because they have no model of how the brain dynamics varies from one individual to another, they have to train these models within each subject, so they can't generalize. But in this particular example, Simon was able to use this model of the spatial function, the spatial temporal dynamics of the brain function in order to be able to generalize these models, which means that you can predict what an individual is looking at in the scanner, even if you haven't seen their brain imaging data before. To do this, you need to use clip contrastive learning again. So you have transformers that are encoding the visual stimulus, the audio stimulus, and the brain activity. You align these. And then what you can do is predict what scenes of the movie they were watching. So because clip is a ranking system, you can show it all sorts of different movie clips, and it can rank which ones it thinks are most likely to be the ones that the individual was watching from their brain activity. So here you're testing on subjects that haven't been seen during training, and you see that not only does it predict the right scene, but the top scenes have the same semantic content. So these are all faces. And the bottom scenes are completely different. So we've got buildings, for example. You can also start to generate frames of movies just purely from the brain activity. Whereas this is quite preliminary work based on somewhat leveraging stable diffusion in a network based on results by Arcelic and Van Rulen. But you can see that, here you've got staircases and it's generating staircases and kitchens. Here you've got Inception, two people walking down a street with cars and buildings. It's the same semantic content in the movie, just being generated directly from the fMRI. So here you're in a position where you can start to use these networks to potentially model different cognitive processes, partly because the attention mechanism itself, you can look at what it's encoding, and it's going to give you some information about it gives you some indication of the cognitive processes or the functional, the brain networks that were involved in encoding that signal. So this is what the model has to this is what the model is focusing on in order to predict the stimuli from the activations. But when you look at it, you see that the attention heads themselves, so during your transformer, you learn different heads, and each of the heads should be modeling a different part of the signal. And each one of these is modeling different recognized functional networks in the brain. So the first 4 are looking at different components of the sensory, motor, auditory functional networks, and the last ones are encoding visual processes. So the model has some inherent, innate model of the cognitive, the cognitive processes. It's in some way encoding the model of the cognitive processes in order to be able to predict this. And while in this case, we already know what processes are involved largely in people watching movies, if you had some sort of cognitive class where you weren't really entirely clear what the cognitive process was, you could look at the attention maps and it would give you some greater way of interpreting or proposing mechanistic hypotheses about what the brain was doing. But at the moment, we have no specific biophysical constraint on these models. You know, this is still purely data-driven. But where I think the direction of travel needs to be going is to embed your models with biophysical constraints or causal models that they will give you back some explanation for what is going on mechanistically in the brain. So there are a couple of different ways of doing this. The first one, which is quite popular, is deep structural causal models. So in this case, you need to train a sort of separate network on the tabular data that's going to learn a directed graph that tells you how different characteristics contribute in order to causally generate some particular image properties. So this was a 2020 paper from Imperial. They validated it on brain aging, where here the model is quite simple. It's just saying that age of an individual and their sex have a direct impact on the brain, overall brain volume and the ventricular size. And that then combined will determine the appearance of the brain. I tend to have a little bit of skepticism about these models because you'd have to have an extremely complicated causal graph to model all the sources of possible variability that would contribute to that contribute to the appearance of any given brain. I therefore tend to prefer the direction of biophysically constrained models or physics-informed neural networks. So here the idea is that you are training AI-driven, so data-driven models, but physics-constrained models that will give you back a mechanistic explanation from what's driving some particular imaging phenotype. So in this example, for example, we can model the biomechanical processes that are involved in brain atrophy during Alzheimer's. So we know there are many biomechanically proposed physics models that explain brain tissue deformation for atrophy and for cortical folding. And what you can do with AI models is you can train your model to embed, so you can train your AI network to behave in biophysically plausible ways by regularizing it with these physics models. So in this particular case, what you're saying is that we're giving the network just the demographic variables. So the age of an individual, their diagnosis and their sex, and some interval between 2 scans. And from population data, we say we expect that these different areas of the brain are going to atrophy by some specific amount, isotropically in all directions, so evenly. And then your biomechanical model is saying, I want you to output deform this brain in such a way that you minimize deviation from this prescribed model. So if this is your prescribed amount of atrophy or growth, if you were modeling cortical folding, and this is your total deformation the network puts out, you penalize the strain energy, so the deviation, the elastic deviation from that model. So you're forcing a physics model And meanwhile, you're training the network with the imaging data. So you want it to deform this baseline scan so it looks as close as possible to the an image at a later time point while staying as true as possible to the biomechanical model. And the advantage of these types of approaches is that they then learn to extrapolate beyond the data regime. So you can prescribe any amount of atrophy as you want, and you can model disease trajectories over whatever time span you want. And it will change the brains in ways that clearly differentiate between healthy controls and disease patients. So you can see that at test time, you're just giving the model the age at which it starts. So that will be corresponding to the original scan, but some interval over which you want to model the brain atrophy. You could change their sex if you want. You can change their diagnosis. And if you do that, you can just simulate how you would expect their brains to atrophy if, for example, they were to convert from mild cognitive impairment to to Alzheimer's disease, and then potentially clinically follow them up to see whether they're following that trajectory.
So do you have a way to embed like some measure of uncertainty that predictive model? Because I guess it works like a digital twin or like a simulator where you just pick some parameters and then it will give you some trajectory of development, but then that's not unique, right? There might be some variability around that particular trajectory.
Yes. So, I mean, the way that I was thinking about it is that these biomechanical models themselves aren't perfect. So any deviation, you know what the ground truth is for these. So any deviation where the model isn't working very well will give you indications that you need to improve on the model. And also, in principle, and there are papers that are doing this that are coming out of Stanford, you can train models to design the strain energy function, constitutive networks. So you can actually learn a better mechanistic model of your problem directly from the data.
So you mentioned the limitation with previous approaches that you did a detailed causal diagram or some model in order to use it, but don't you also need some reasonably accurate physics model here, or so you can use it to inform the kernel, but otherwise it might be misinformed?
Yes, exactly. So I think in terms of brain, for atrophy, this works quite well. For cortical folding, the cortical folding based biomechanical models, they generate brains that look somewhat human-like, but not exactly like the individual. So there's a lot of space, but I think that's where I'm saying, you have the space to learn from the data because it's trained on data to improve the model. Whereas I see what you're saying, but with a causal model, what would be your causal, how would you even go about deriving a causal graph of Alzheimer's? So it's definitely a lot more complicated than the ventricles get bigger and the brain atrophies. So you'd have to have, they are trying to include, ApoE status, blood measures, things like this. But actually what's really, unless you do it by separating out in an ICAM-like way, the variation that you're trying to model from the variation that you're not, most of that variability has nothing to do with these sort of arbitrary tabular variables you're putting in. And certainly from a developmental perspective, a lot of these measurements, if you put in, for example, cognitive tests, like the MMSE from Alzheimer's, they're so noisy. Like somebody will do the test 2 different days in a row and they'll come back different. Like we can't tractably train a regression network on any of those, let alone a more complicated causal model. So at least with a biomechanical model, you have an ability to tune how much you force it to follow the model and how much it follows the data. So it's a regularizer, it's not a hard constraint. And with that potential that you can then, from the data, learn a better model is kind of my position on it currently. But at least it gives you some form of framework to perform hypothesis testing, so you can either provide a hypothesis and test it, or you can use the whole framework to generate entirely new hypotheses.
So it depends a little bit on how much you know, basically, and if you only know like vaguely like satisfy this, you can just use it as regularization, but don't want to enforce it. by building it into the causal background, so, or something.
I think there are papers that do both. Yeah, they use causal models to learn the physics parameters, and then you can do both, which does sound quite interesting. But these physics models, they're not. I mean, they're not that far. So the image registration tool that I created for surface registration, it has a biomechanical regularization in it. So longitudinal registration and deformable modeling, biomechanical modeling are quite paired. And you can learn just from data. You could try and learn just from data, and people do have generative models that learn deformations instead of just to generate it. And then they can model aging in that way. But I think in the space with brain imaging, or in general biomedical imaging data, where you've got you're working in a relatively low data regime, it makes sense to put in what you know. So to add in biophysical constraints. Yeah, so that's it basically. So I think if there are any main takeaways that I would suggest from this talk, is that in my experience, if you want to be going about building AI tools that leverage medical imaging data, we don't have anywhere near the amount of data that they're using to train natural language and natural image processing networks. And yet arguably the data is way more complicated. So you can't, oh, hang on, going too far. You need to bring down the space of variability you're trying to model through embedding your understanding, your current understanding of the neurobiological problem in this particular case into your network design in order to be able to learn more tractably and generalize better. And from where I've got to with neuroimaging, I would say that means that you need to understand that most of the variability in your data has nothing to do with the phenotype that you're trying to model, that you need to define normal relative to an individual and not relative to an average population, that the cortex is inherently a surface, and for many problems it makes more sense to model the data on the surface than in the volume. And that if we want to do better or make really good use of imaging data, especially like these advanced 7T MRI scanners, which are never really, no near future are going to be used for clinical practice, then you really have an opportunity here to use this imaging data and to improve your mechanistic understanding of what's driving the imaging phenotype. Thank you all.